Hello, welcome to a new video tutorial on anomalous secondary growth in boerhaavia. We know that boerhaavia is a plant belongs to the family Nyctagenaceae, and the stem of boerhaavia shows a secondary growth which is somewhat different from an ordinary secondary growth. That is why it is called anomalous secondary thickening. Let us try to understand what are the main features of this secondary growth. Before going to study the details of this anomalous secondary thickening, we have to understand the basic structure or primary structure of the stem of boerhaavia diffusa. Look here, you can see the anatomy of boerhaavia stem. In this diagram, you can see an outermost layer. That outermost layer is called epidermis, and that epidermis contains, this is called epidermis, and that epidermis is a single layer. It contains a wax-like coating called the cuticle, and also presence of stomata is there. And one more thing is that epidermis is covered by wax-like coating called the cuticle. So 4 points: single layer, contains stomata, contain multicellular stem hairs, and a protective coating or mucilaginous coating or wax-like coating called the cuticle. And followed by, when we move into the innermost region, inner to the epidermis, you can see cortex. That cortex is divided into two regions. Outer region, that is called hypodermis. That outer region is made up of 2 to 3 layered collenchyma. That outer region is made up of 2 to 3 layered collenchyma. And followed by parenchyma tissue. So cortex is divided into two. Outer cortex is called collenchymatous hypodermis, and inner cortex is called parenchyma. And that region is followed by an endodermis, single layered endodermis. And inner to endodermis, you can see, inner to this endodermis, you can see few layers of pericycle. Normally, that pericycle is made up of parenchyma tissue, whereas at some region that parenchyma tissues are replaced by few sclerenchyma tissues. That is pericycle. And when we move into the innermost region, you can see inner to that pericycle, you can see lot of vascular bundle. That vascular bundles are arranged in 3 rings. This is the first ring. You can see this is the first ring. This is the second ring, and this is the third ring. So, in the main features of this boerhaavia stem is that presence of 3 layer of vascular bundle. Among these, innermost ring is occupied by 2 large vascular bundles. And middle ring is occupied by 6 to 14 vascular bundle. And at the periphery, the third ring, outermost ring, occupied by approximately 20 vascular bundle, 14 to 20 vascular bundle. And one more thing is that smaller number of vascular bundle is present at the innermost region, and they are with larger size. When we compare innermost ring and the middle ring, you can see number is 2, here number is 2 to 6 to 14. But the difference is that here the vascular bundle is large, here the vascular bundle is small. And when we move into the third ring, outermost ring, here the number of vascular bundle is more, but the size of vascular bundle is less. So when we take the size is larger for inner, then larger for middle, then larger, then comes outermost. So smallest one is this smallest vascular bundle present at the outermost ring, largest vascular bundle is present at the innermost ring. And one more thing is that inner ring of vascular bundle and the middle ring of vascular bundles are occupied at the medullary region of this term. That is why inner vascular bundle and the middle vascular bundles are also called medullary bundles. And when we move into the arrangement of this vascular bundle, you can see protoxylem, that is primary xylem is directed towards pith, and phloem is directed towards the cortex. That is why the whole xylem is whole xylem is enda. And one more thing is that you can see vascular bundle, you can see xylem and phloem. In between the xylem and phloem, you can also see cambium. That is why this vascular bundle is open. That means it shows secondary growth. It contains cambium in between the xylem and phloem. That cambium is called intravesicular cambium, or we can say vesicular cambium. So the vascular bundles are open and they are arranged side by side. That is why we can say that it is a conjoined vascular bundle and open vascular. Conjoined and open type of vascular bundle are present in the whole structure. If you take any vascular bundle, you can see they are all conjoined and vascular because they are arranged side by side, and vesicular cambium or intravesicular cambium are present in between these two, xylem and phloem. Now let us move into the secondary thickening, which is going to happen here. When we take the outermost ring of vascular bundle, you can see vesicular cambium. When we take the inner and middle ring of vascular bundle, you can also see vesicular cambium. So in all these cases, vesicular cambium is present. But when we take the outermost vascular bundle, you can also see intervesicular cambium. You can also see intervesicular cambium. The main features of this boerhaavia stem is that outermost vascular bundle contain both vesicular cambium and intervesicular cambium. Here I am drawing the intervesicular cambium by using red marker pen, so you can easily able to see. The main features of this boerhaavia system is that its outermost vascular bundle contain both the vesicular and the intervesicular cambium, whereas the middle and the inner vascular bundle contain only vesicular cambium. So majority, in 99% secondary growth takes place in this outermost vascular bundles. So let us move into the secondary thickening. And one more thing is that this vesicular cambium, black colored vesicular cambium and red colored intervesicular cambium united together and form a cambial ring. And this cambial ring is made up of only fusiform initial. That cambial ring made up of only fusiform initial or fusiform cells. Whereas ray initials are completely absent, whereas ray initials are present in the vascular bundles of a normal dicot stem, but here it is absent. So the main features of this boerhaavia stem is that its cambial ring lack ray initial, and this vascular bundle is made up. So this cambial ring is made up of vesicular cambium and intervesicular cambium. Among these, this vesicular cambium produces a group of tissue towards outside and inside. The those group of tissue which is produced towards outside is called secondary phloem, and those tissue which is produced towards inside is called a secondary xylem. It is similar to normal dicot stem, but the difference is that the intervesicular cambium not shows the activity like that of vesicular cambium. Intervesicular cambium produces a group of parenchyma, loosely arranged parenchyma tissue towards outside. Like that, it also produces parenchyma tissue towards inside. But here, vesicular cambium produces secondary phloem towards outside and secondary xylem towards inside. But intervesicular cambium not produces secondary phloem towards outside and secondary xylem towards inside. Instead, they produces parenchyma tissue towards outside and inside. Among these, those parenchyma tissue which is produced towards inside gradually converted into dead cell by the heavy accumulation of secondary metabolites like lignin, and they gradually converted into sclerenchyma tissue. Such group of cells or such type of tissue are called conjunctive tissue. Such type of tissue is called a conductive tissue. So in simple terms, this interspecific cambium produces conductive tissue towards inside. Like that, it produces live parenchyma tissue towards outside. Whereas specific cambium produces secondary phloem towards outside and secondary xylem towards inside. So finally, when we look at the structure of this secondary growth, when we look at the structure of this boerhaavia stem after secondary growth, you can see discontinuous layer of secondary xylem, because here it produces secondary xylem, here it produces secondary xylem, and then it produces conductive tissue, then secondary xylem, then the conductive tissue. So we cannot able to see a continuous layer of secondary xylem because each secondary xylem is interrupted by conductive tissue. That is the structure which will generate here after secondary thickening. Okay, this is somewhat different from an ordinary secondary thickening, because in ordinary secondary thickening, the newly produced secondary xylem is a continuous layer, but it is a discontinuous layer due to the presence of conductive tissue in between each adjacent secondary xylem. This is the secondary thickening which was happened here. And after few days, after few months, the nature of this cambial ring is not like that of an ordinary dichosome. Here gradually the cambial activity or division activity of this whole cambium gradually diminishes or it decreases. So after a particular period of time, this cambial ring cannot able to produce a secondary xylem, secondary phloem, conjunctive tissue. So finally, this cambial ring gradually diminishes, and a new cambial tissue will emerge instead of this cambial ring, because this cambial ring loses its division capacity. So instead of this cambial ring, the stem produces new cambial ring from the phloem parenchyma cells, from the newly produced phloem parenchyma cells, because this specific cambium produces phloem tissue towards outside. That phloem tissue contains pseudotuberum and companion cell phloem parenchyma. That phloem parenchyma produces a new cambial ring over here. It produces a new cambial ring over here. And the new cambial ring again produces secondary phloem towards outside, secondary xylem towards inside, and like that conductive tissue towards inside, and also phloem, xylem, and also parenchyma towards inside. So this is the mechanism which was happened in the boerhaavia stem. And after a particular period of time, again this newly created cambial ring loses its division capacity. And finally, again the newly produced phloem parenchyma tissue create a new and third cambial ring. So the process of this repeat up to 3, 4 or 5 times, so that we will get a continuous ring of 4 to 5 ring of secondary xylem and secondary phloem here. This is the secondary thickening which is taking place inside the dicot stem, that is boerhaavia stem. Due to this abnormal nature, this secondary thickening is called anomalous secondary thickening. Okay.